So on this module 1.4, we will now talk about all these other plug-in energy terms that actually influence the shape of the cells. Okay. So again, zone logistics, you never use, but you are free to use these buttons of yes, no, go slower or go faster. Okay. If you please use the chat to answer my questions and say yes, no, makes sense, does not make sense. If you need help, let us know, and some of the person on the team, we can come and help you. Okay. So we all be doing all those pulls as you go along to know if you can proceed in the exercises, and everything is going to be made available to you by the end of the course. I know that you have an incomplete set of slides from the previous module on the student materials. I will upload the complete one as soon as this is done, okay? I promise. So in this module 1.4, I'm going to give a motivation, which is cell shapes, and then we do the step-by-step exercises in the same format as we did before. Okay, so the motivation here is very simple. Cells do not look alike. The cells have very different shapes. Okay, so here is a C, and they can vary a lot. The size varies a lot, the shapes vary a lot, because each cell is doing a very different function. Okay, so these are brief review of all different cell shapes. And before we talk about epithelial cells and mesenchymal cells, we kind of said that all the epithelial cells look like cubes. That's not actually true. The epithelial cells can have very different shapes. They can be squamous, like a pancake, but still strongly connect to the ones on the side. They can be cuboidal, as said before. They can be very long and columnar. And they can have these other types of shapes. Some can be a scutoid or trapezoid, and well, you can see from this picture that very different shapes. Well, when you go to mesenchymal cells, there is also a very different types of cell shapes. Here's so all the pictures on the top is from the same embryo, from the same tissue. It's all mesenchymal cells in the chicken limb bud mesenchyme, and they can have this very different shape. They can be very have all these protrusions, filipodia. They can be like just polarized one direction, polarizing both directions, or polarizing many different directions. And here on the bottom you see fibroblasts, neurocrest cells. Okay, very different shapes. And then if you look at this and look what you had before, many different cell shapes. And the question is, okay, if you have a specific cell in mind that you like to model in CompuCell, the question is, how can I tune my parameters to make the cells look more like this or more like this? I already tell you that it's very hard to make those cells to look like this in CompuCell. You will need a lot of pixels. You probably need to actually break your cells in small compartments. But you can, there are things that can be done. And the idea of this module is to show all the parameters, or the basic parameters in CompuCell, that can actually have an influence in the shape of the cells. So in the last module, we already see that there are different ways to model cell behaviors and cell shapes. This is a simulation where the behavior of the cells were more or less the same. They are fragmented and spread around, but the shapes of the cell were drastically different from one to the other, right? So that simulation can also be a motivation. Okay, what are the parameters in CompuCell that actually influence cell shapes? Okay, so now let's go to our step-by-step exercises. Okay, so the first thing we want to do is the same as before. We want to set up a basic simulation that we use to do our exercises. So again, please go to Twedit and click on the menu, Compensating project, and then select New Compensate 3D project. Okay. Okay. So once you get there, you open up the wizard. Okay. So when you say wizard, what it means by this sets of screens at the beginning when you set up a new cell simulation, and then please give the name of your simulation. So here I gave module 1 underscore 4 underscore cell shape. Again, please no spaces, no dots, no slashes, or question marks or exclamation marks in your name, just letters, numbers, and underscores if you wish. Okay, so give the name of simulation, Python plus XML. Here in this simulation wizard, you can leave everything as it is, okay? And just hit continue. Now it comes to the part where you have to actually give a name for your cell. You'll be doing a simulation with just one cell, and again you have to give a name. That's one of the parts that I do not like. But here I just gave a name of test cell. Okay, so with the cell we will be testing all these parameters on it. So again, remember you have to type the name of the cell and then hit add, and make sure that the name of our cell, whatever it is, appears here on the table before you hit continue. Okay, so give your cell a name, whatever you may want. You can just use test cell as I give here, and hit continue. And now this will lead to our another window called chemical fields. Ignore it and just hit continue. Okay, we're not doing chemical fields today. And now we go to the very important window, which is the cell properties and behaviors. And now you want to select many things. So I want you to select contact, volume flex, surface flex, and length constraint. Okay, make sure you have those 4 things selected. It does not hurt to have cell center of mass also selected, but you absolutely need the contact, volume, surface, and length. Make sure you have all those 4 selected, and then you hit continue. Okay. Everybody's on board? Okay, so let's continue, and then congratulations, the configuration is complete. Once we achieve here, hit done, and then we can be ready to start exploring the simulation. Okay, so hit done, and again, let's not look at the code at the beginning. Let's just open the simulation in the player and see what we get. The player crashes when I want to run this model. Okay, if someone in the team could help Farnush with her model, I will appreciate. Okay, is this crashing for everyone, or it's running okay for you, for the rest? Okay, it's running for most of you. So for those of you that did not work, please raise your hand, and someone from the team will get in contact with you and probably go to a breakout room and check what went wrong. Okay, the issue. So if you run simulation, what you get is something that looks exactly equal to what you had before, right? It just creates a group of cells that rounds up. Okay, No surprising, because you actually did not did any configuration in the simulation. What you want to do is actually now go to the XML and change the simulation to have only one cell at the beginning. Okay, so yeah, but you like to simulate single cell, right? So how to go back to the edit? I go back to the edit by, so on the comp cell player there is an icon which looks like a piece of paper with a pencil. You click there and you go back to Twedit. Okay. Your cells are a little bit elongated. Yes, 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 that's true. Okay. Anyway, the simulation right now does not do what you want. Let's go now to the code, to the XML, and actually configure the thing to do what you want. So, oops. So now let's go back to Twedit and the code, and right click here on the name of our comp cell simulation, module 1_4_ cell shape, Compensating to open up this and select XML, or click here on the XML to actually see the XML code. Okay. Okay. Yeah, the cells are doing lots of different things because all the plugins when selected, they started on. Okay, and we do not want to export them all at once. You want to export them one at a time, right? And you also want to export that in one cell instead of many cells at the same time. Okay. So if you want to go to the breakout room for help, please send a message and someone will assist you. Okay. So let's go to the XML code. Do this part here. Click on XML to actually open it. And now the first thing that I would like you to do is actually to change the dimensions of your system. Okay, so under the pot section, okay, which should be around line 14, it actually should be exact line 14. That's where you have the dimensions of your system is set up. I want to change the X and the Y from 256 to 100. Okay, so please make sure you have your parameters just like as I have on my screen. X equals 100, Y equals 100. Okay. Everybody's following? If so, now let's change the initial condition of the cells. So let's go out to the end to the XML code where we have Uniform Initializer, okay? And instead of having a bunch of cells, I want to just draw one cell. So the way that the box initializer works is that you set 2 points, a box minimum and box maximum, and from those 2 points, the computer draws a square and fills this square with cells of a given width. Okay, so right now it has from 51 on X and Y to 204 and 204 on the X and Y, and it's filling with cells of type your name of your cell, and those cells are 7 by 7 pixels in width. Okay, so X and Y are the same. What I want you to do is to instead change those values and make your X and Y start at 40 on line 61 and make your X and Y end at 60 on line 62. Okay, so now you're drawing a 20 by 20 box, okay, right in the middle simulation. And okay, so change those 2 lines, 40 40 on the top, 66 on the bottom. But you do not want to fill this box with 7 by 7 cells. What you want to do is to fill this box with a single cell. So to do that, you just specify your cell to be exactly equal to the width and height of the box. So make width equals 20 and now you're drawing a 20 by 20 region where you instruct CompCell to add a 20 by 20 cell. So your simulation now you have just one cell of 20 by 20. Okay, does this make sense? So there on the slide are all the previous steps in case you got behind. But now I'm gonna step on the next slide. How to make comments in XML file. You can look here on line 59. Okay, this is a way to make comments. You make this sign here and an exclamation mark, 2 slashes, and then you can add a comment to it. Yeah, thank you, Sluka, your comment there. Yeah, that's a good practice to make comments on your code.
One thing to beware of, though, with XML is that if you comment a comment, it will crash. You can't put a comment inside of a comment.
Good reminder. Yes, thank you, James. Okay, so assume that everybody's on board. Saved simulation, remember? And let's save simulation. You can go to the menu and save, directly hit the saving icon, or hit the shortcuts Control S if you're on Windows, or Command S if you're on Mac. You can tell that I'm on a Mac here, right? So save simulation, and I'll play it and tell me what you get. Okay. Save, play, and do you get what you expect or not? Okay. So when you play simulation, you should see a initially square cell of 20 by 20, exactly what you wanted, okay? But it quickly reduces its size and looks very funny in the end. It looks very rough, okay? And very kind of elongated, right? So why does this happen? Okay, I already have the answer there. Yeah, it started getting smaller, right? Why is it getting smaller if I set myself to be 20 by 20? Because, yes, the target volume has not been changed, right? So we forgot to do this. So it doesn't matter what the initial condition of your cell is. If its target volume is different than it is, it's gonna go towards that target volume, okay? And it does it very quickly, as you can tell. Now, why does the cell looks kind of elongated? Does not look round. Why it looks elongated? Because the length constraint is there. Okay, we did not turn it off. It also looks very rough, right? It seems that it have more surface than it needs. Okay. Again, we set the wizard to add a surface constraint, and we didn't do anything. We just leave it there. So it still has a surface constraint happening. So there are too many constraints working on the cell right now. Okay. So we cannot study all of them simultaneously. What you want to do now is to just turn all of them off and leave only the volume plugin to begin with. Okay, so that's exactly what you do. So yeah, so in the current simulation you have 4 plugins turned on at the same time. The cell is over constrained, and it's there because you just tell the wizard to be there. Okay, so let's all these plugins work in a very similar way, except for the contact energy. The volume, surface, and length plugins, they have this shape which is look like a spring, as we talked before. Okay, so volume, surface, and length string, they have this format where is whatever property value that you have minus its target volume. This difference is squared, okay, times some kind of strength or lambda value or the strength that says how strong this constraint is on our cells. If this value is low, it's okay to deviate from this target value. If it's very high, you restrict, you want to be very close to this target value. All right. Okay, so this is exactly the format of the volume, surface, and length string. They all look exactly similar. Is a strength of the constraint, the current value of that property, and the target value that you wish this cell to have. All right. And then this is how, when it's on the target value, the energy term is 0. When it deviates from the target value, either higher or lower, you get an increase in energy, and that is penalized in the way that CompCell works. Good question. What does volume mean in 2D? It's just okay, in 2D you don't really have a volume. What you have is an area and a perimeter, right? So in a 2D simulations, the volume of the cell is the number of pixels or its surface or its area, as you see from the top, and its surface is actually the perimeter of the cell, as you see from the top. Okay. When you go to the 3D simulations, now the volume is the number of pixels or voxels. Voxel is a pixel in 3 dimensions, right? And that is the actual volume. And the surface is actually the number of is actually a surface, all the boundary surface of that involves your set of voxels that make up your cell. So in a 2D simulation, surface means periphery, and volume means area. Okay. So that's a very good distinction. So we'll be only doing 2D simulations today. Okay. Could someone share the link for the live video on YouTube? Okay, thank you. All right. So let's continue. So before we can do anything useful, let's turn off all the plugins that will not be used at the moment. So please go to Twedit, double click on module 1_4_ cell shape on our CompCell3D project on the left panel, so you can expand the contents of your code. And let's click on the XML to actually see the XML code in front of you. Okay. Once you get there, let's first turn off the Surface plugin. So remember, the Surface plugin has this shape here, and this is the place in the XML code where it specifies the Surface plugin, and is listed by cell types. Is only have one cell type simulation, and here are the two parameters: the strength of the constraint and the target value of the surface in this case. So we want to turn it off, and the way to turn it off is just making this 0. When you make this 0, does not matter what's happening to the cell surface. It does not contribute to the energy of the system, so that's the same thing as turning off the plugin. Okay. And now, so with this we turn off the Surface plugin, and the next thing I would like to turn off will be the length constraint plugin. Okay. So you do the same thing. You'll be in a different line of the code. It should be around line 48. Okay, and if you go below, again, cell type, strength of the constraint, and the target property of your constraint. So let's set the lambda length equals to 0, and the target length, you can leave that number, it doesn't matter. Once you set this to 0, this can be whatever you want. This is not going to be read by the code. Actually, it's read by the code, but does not contribute to the dynamics of the cell. Okay. So, are you all with me? Yes, thank you for answering. So now let's save this code now with the two plugins turned off and play the simulation. So remember, we have to hit this button or go File, Save to save it. And to play simulation, you have to either right click or double click on the name of the module, hit Opening Player, or just hit this. And you should now see a simulation that looks very different than what you had before, right? So now you should see that the cell that, oops, it still decreases its size, right? But it has a different shape now. Do you all get something similar to this? Yes or no? Yes. So we fix two things, but we forgot to fix the target, the volume of the cell, right? We took out the length constraint and the surface constraint, so you see that the cell looks very different from what you had before. Before it was kind of elongated and very rough. Now it looks a little bit more round, okay? But because you'll be doing a whole module on the cell shapes, I don't want to do all the exercises on a very small cell. I want to do the exercise on a very big cell, so you can appreciate the effects better, okay? Your cell's not decreasing size. Maybe you fix it, okay? So what you should do in the code, you want to turn off the surface of the cell, the lambda, the surface plugin, and I want you also to turn off the length plugin. Okay, so we turned off the two plugins, but we did not touch the length constraint. So the cell still starts in a big square, 20 by 20, but it still decreases and happens super fast. If you could not see it, stop simulation, and instead of hitting the play button, you hit the button next to it, which is the one with the circle inside, and then you can start the play simulation step by step, and then you can see those intermediate states. Okay. So let's fix this. Let's make the cell to want to have that 20 by 20 volume. Okay. So to do this, well, the reduction volume is still happening because we did not touch. There's a discrepancy between what you set the initial cell to be, right? That 20 by 20 set of pixels, but the target volume is still set to be 50, which is the default value that the wizard gives you at the beginning of the simulation. So what you want to do now is actually make this target volume to be equal to 20 by 20, okay? Actually, what you want to do is now, this will be our first exercise. What happens if you change the target volume of the cell? Okay. You have several cells set 1 because you probably did not change the uniform initializer. So look at your code around line 57. Make sure that your box coordinates, which you use to draw your cells, is 40, 40, 60, 60, and make sure that the width of the cell that you're going to draw inside this box is 20. Yeah, so make sure you have those values here in your XML code. So if you're already there, please try now to do some simulation with higher and lower values of the target volume. Okay. So yeah, so that's basically what I want to do, which is the result of 20 by 20, right? Okay, good question. What's the difference between length constraint and surface constraint? We will get there in a minute, okay, or in 20, 25 minutes, but we'll talk about it. Okay, so now if you change your target volume to 400, the cell now keeps its volume. Of course, it does kind of change its shape. Initially, it was a square, now it's round. This is happening because you not only have the volume plugin turned on, but you also have the content plugin turned on. So there is some kind of cost for the cell to have surface with the medium. Okay, so the cell a square is not very good, so it wants to round. Okay. So one of the exercise, the first exercise I want you to do in this module, okay, now let's play with this target volume, make it bigger or smaller, and let's see what happens. Okay, so please start to do this now and try higher values and lower values, and let me see how it goes. Okay, you right hit something that we will talk about. For this one, it's very nice to do that actually on the player. You can change the value of the target volume direct on the player, and you can see the cell growing and shrinking, growing and shrinking. Okay. So while you do this exercise, I will try to upload this set of slides to the studio materials right now, so you can actually go back and catch catch up with the rest of the class. Okay, so when you play with the target volume, you what you expect to see is exactly like this. If you make the target volume smaller, you get a smaller cell. If you make the target volume bigger, you get a bigger cell. Not much surprise. You can also try to set the target volume to 0, and what you get is exactly what your colleague said. If made to 0, the cell disappear, and that's an efficient way to uh, to to kill the cells in your simulation. So let's say the cell simulation should die because of so and so and so. You can set the target volume of the cell to 0, and the cell will shrink and disappear. Okay. The surface plugin by making lambda surface equals to 0. Okay. By the way, for the person that crashed the course and was behind, I just uploaded the 2 PowerPoint slides to the studio material folder in the Google Drive. You should be able to go there, download, and open it. Okay. Yes, the volume changes fast. It's not instantaneously, but it can get bigger and smaller very fast. But that happens in this simulation where you don't have other cells compete with, right? Once you have a cell embedded surrounded with other cells, the change in volume can take a little bit longer. Okay. So the current parameters, okay, we turn off the surface and length constraint plugins, and we are just now exploring the effects of the target volume. So we're basically doing we are exploring those parameters on line 27 to 29 in the XML code. Okay. So if you got lost a little bit on track, the slides have been uploaded. You can download and see them by yourself, the code. So good mind by James, you want to kill a cell, you not only set the target volume to 0, but you also want to make the lambda volume very high, so the cell really wants to shrink and disappear. So just setting the target volume to be is not guaranteed that it will disappear. You may also have noticed as you are playing with this that when you set a target volume lower than 30, the cells also disappear. Okay, if you set for 20 or 10, they also disappear. There's a reason for that. We'll talk about that later in this module. Okay, there is a good reason for that. You understand that, but let us get there. You'll get there in an instant. Okay. So for the remaining of the module now, I want you to set the volume to 1000. Okay, so I want you guys to have a very big cell. Okay, and let's now explore the effects of the lambda constraint. Okay, so let's make the target volume equals to 1000 pixels, and let's well, you can start by setting the lambda volume to 2 and see what happens. Okay. So when you set to 1000, the cell grows a little bit, but it stays like that. And you can see that the cell is not frozen, right? It has this bigger shape, but the pixels around the cell are fluctuating, right? The cell is still able to move. You have some membrane ruffling of the cell, so the cell looks healthy in a way. Now, okay, now you have the simulation running. Let's try to make your lambda volume from 2 to 50 or even higher values, and tell me what you expect to happen if you make the target volume, the strength of volume higher, and why it would happen. Please, before doing simulation, tell me on the chat what will happen if you make your lambda volume to be higher. You can also unmute yourself and tell me what will happen. The two answers are correct. Less fluctuation, converge to target faster. Yes. Oh, less variation, okay. Okay, good guesses. Let's try the simulation, see what you get. Okay, so this is what happened on my end. Okay, yes, the cell does grows faster. Okay, it was 20 by 20, now wants to be higher. Okay, Uh, but you see that after this very fast increase, the shape fluctuates much slower than before. So this is a simulation of targetVolume equals 50, and I took the screenshot still at the beginning of simulation. I did not have time to round up. So you see that it does not look, the cell looks a little bit less dynamic than you had before, right? And the reason that this happens is written here on the slide, is that once the cell achieves its target volume, right, is already happy, and it does not want to accept other spin flips, or pixel copies that will change the volume. So once the cell went from this square of 400 pixels to this shape that actually matches 1,000 pixels, it needs a little bit more pixel copies to round it. Okay, but the problem is that all those pixel copies that are left to round the cell will be penalized, because they mean that you'll be momentarily deviating your current cell volume to a higher or lower volume to achieve that rounding. And when that this lambda volume to a number that's too high, this will be penalized. So you're gonna slow the cell. You'll be kind of freezing the cell when you do this. Okay, so if you set your lambda volume to 50, the cell can still round, but it's gonna take a little bit longer. Does this make sense? Yeah, so the cell grows faster towards its target shape, but once it gets there, it's harder for it to evolve its shape. Exactly, Tommy. Is actually that's exactly like that. It is a deeper energy well. Okay, so it is harder to fluctuate your energy around. Okay, if this makes sense, let's proceed. Okay, if you have not, there should not be this not there. Try to make lambda volume equals to 100 and see what happens. What do you expect to happen if you make it the lambda volume equals to 100 or even higher? You can try 1,000 and tell me what the result is. No, keep the target volume to be 1,000. But now let's make the lambda volume to be 100 or 200 or 500. Janus got a more rigid cell. Actually, maybe you not get rounded at all. Okay. Yeah, the cell becomes static, right? Because exactly, it denies most pitch copy attempts. Okay, it grows a little bit at the beginning, right? Right. So if you stop simulation and play it again using the step function, one Monte Carlo step at a time, you see that it grows, and once it reaches its target volume, it's gonna stay there and not change. Okay, so setting the target volume, the lambda volume to be a super high number is a very effective way of freezing the cell in your simulation. Sometimes you want to have some cells in your simulation that do not move, that do not change shape. They are more like the part of the environment of your simulation than what you're actually simulating. You can enforce that by either right at the beginning of the wizard telling the wizard to freeze those cells. There is an option freeze, that's what it means. But if you want to do that in the middle of simulation, you can also do that by just making your tag lambda volume to be a very high number. You make it 1,000, a million, and that will freeze your cell. Okay, so why this is happening? Because again, once if you make your lambda volume very high, you're basically penalizing all the pixel copy attempts that will deviate the cell from its current target volume. Okay. Okay. So 100 effectively frees the cell in these current simulations, but you may be surprised that in another simulation that you do further along, you set your lambda volume to 100 and the cells still move. Okay. Then you may wonder what happened. Well, again, what matters is not the absolute number of the target of the constraint strain, is its vol is but its value compared to the other parameter values and also the temperature of your system. Okay, if you set your temperature of the system to be at 1,000, having a lambda volume of 100 will not freeze your cell. Okay, so here 1,000 is already enough to freeze your cell because the default temperature in the comp cell simulations when you start them with the wizard is 10, and 100 is very bigger compared to 10. Okay, so keep that in mind. Okay, you have always to see how this plugin parameters of this plugin are with respect to the other settings of your simulation. Okay. Oops. So let's try to do this. Let's put this on the test. In the current simulation, the temperature has a default value of T equals 10. Okay. You can check this by looking at Twedit and look at the pots section of XML, R10. So let's put this to the test. Let's see if the structure here is telling you the truth. So let's keep the lambda volume to 100 and set the temperature to 102. Okay. So you might expect the results to be similar to simulation with 10 and 10. And now I want you to tell me, does this actually happen or not? Okay. So please try to do this and tell me, should you get the same results? Yes, no? Or maybe better, try the simulation, tell me if you got what you expect or not. Remember, temperature here does not mean temperature in Celsius or Fahrenheit or Kelvins. Temperature here is more of an inherited term that actually means the amount of noise in your simulation. Okay, I don't know how to pronounce your name. The cell is not frozen anymore. Yes, but do you get exactly what you had before with 10, J, with lambda 10 and T equals 10? Or do you get something that looks different? It's not frozen, but it's not the same.
Yeah, it's not frozen and it's fluctuating with a higher pace.
Okay.
It's continuously fluctuating. Before that, it was frozen.
Okay. Okay, good. Thank you. So what's happening? Should not you expect the same result, but it didn't actually happen. What are we missing in your logic here? Very good, Stephen. So read what Stephen said. This is because the temperature is also affecting the contact energy. Okay. So this is what happened. This is what you had before, a cell that grows very quick but becomes freezing. Then increase the temperature and now the cell, okay, it fluctuates, it's not frozen anymore, but it was very rough. And the reason why it was still rough is because you do not only have volume and temperature in your simulation, this simulation also have the contact energy. Remember, we did not turn that off. So what happens when you make the volume, lambda volume equals 100 and the temperature equals 100? and you do not touch the contact energies, is that you decrease the relative contribution of the contact energies to the evolution or to the dynamics of your simulation. So making those two parameters, lambda volume and temperature, high is the same thing as keeping them as they were before and decreasing the contact energy of the adhesion between the cell and the medium. Does this make sense? Now, because the relative contribution of the contact energy is lower, the cell does not care much about its surface area with the medium and becomes rougher. So setting both the volume constraint to be high and the temperature to be high is the same thing as telling the cell I don't care more about the contact anymore. And so that's the same thing as turning off the contact energy. Does this make sense? What happened in simulation? So, yes. Okay. So let's now put this to the test again. Let's redo the simulation with lambda volume equals 100, t equals 100, and now let's make your j between medium and your cell to also be 100. So let's try to recover behavior by changing j medium and j test. So please do that and let me know, see if now it works. Yes, there is a find function in Twedit. Okay, so this is what I ask you to do, okay? And so this is what you had before. Right. Oh, sorry, it's wrong here. This should be a 10, right, on the first panel. Oh, no, that's what we did. So we first, so I got lost in my own slide. So it turns out that a set of values of 100, 100, and 100, so the three parameters that we have in our simulation, is equivalent of the same set of values to be 10, 10, and 10. Okay, so if you raise those three parameters by the same factor, there's two simulations here and here should be exactly the same. Okay, so here you have the simulation where you just increase the lambda volume, the cell grow and freezes. On the second panel, we try to compensate by just increase the temperature. The cell is no longer frozen, but is very rough. But then we also increase the contact energy between medium and cell, and now all of them are about the same level. Actually change this shape. Okay. So is this making sense? Okay, James Luca has a very good point here. In most compensatory runs, you don't want the energy minimum of the system. You want a trajectory in space and time. Okay, so because biology never operates at the global energy minima, because that's death. Okay. Okay. So I know that this may seem a little bit boring, okay, because we are not doing the crypt, we're not doing the angiogenesis, we're not doing the cancer simulation, we are not doing anything that seems to be publishable, right? But these are the very basics of the computational simulation, the cellular plots modeling, okay? And if you have a good foundation here, everything should be much easier to follow later. You're gonna really master the new tools that we're giving you in the rest of the week. Okay. So, is there any questions at this point, or can we proceed?
Excuse me, can we assign different value of temperatures for different cell types or cells?
Yes, yes, you can. There is a way to do it. Now I forgot the exact way, but there is a way to do it, and you do that from the. You can do that from the Python code. I think you can also do that from the XML code. It's not something that I use myself, but there is a way to do this. A temperature for each cell type.
I mean, dynamically, for example, in this type of that, whenever we code in some specific circumstance, we change the temperature according to the, the situation.
Yes, there's a way to. All the parameters that you see on the XML code, you can access them from the Python level and change them during the course simulation. Okay, it can even be made a function of other things. So there is a way to change any of the parameters in the XML code from the Python level and during the course simulation. Okay, we'll talk about that as we advance the course. But yes, there is a way to do it.
Great, because I have the same question for the contact energy. For example, we assign a specific value. I was thinking that how we can change it. Previously, I used like contact adhesion flakes in order, define some molecules, then change the densities of those molecules in order to have different adhesion energy. So I was wondering if we can have contact energy directly with this plugin. We can have it. So
No, the answer is no. If you want to change cell by cell, you have to use the molecular species specification.
you mean the adhesion flex plugin?
That's right.
Okay, so with this contact plugin, it is not possible, right?
No, contact plugin only allows you to define energies by cell type, not by individual cell.
Okay, thank you.
Yeah, the contact plugins by cell type.
Can I just add something?
Yes.
We will show you how to set specific energy, contact energy values for different cells of the same type. Okay, so you can let's say clusterize your cells, group them in clusters, and say, oh, this cluster behave differently from this other cluster, and they are the same cell type. You can do this, and we will show you how to do this in day 3, okay?
Okay, perfect.
Okay, so let's continue, and now let's explore another plugin that affects the shape of the cells. Okay, so and one of these other ones is the contact energy. Okay, so let's explore how the contact energy affects the cells. Okay, so so before we explore the contact energy, let's reset all the parameters of simulations back to the default values, which is temperature equals 10, target volume equals to 1000, lambda volume to be 10, and the J between medium and test cell to be 10. Okay, so let's do this, And once you set up all those parameters back to the original values, let's now play with the addition between the cell and the medium, okay, which is the only addition energy that plays a role in the simulation. And I want you to make those values even either higher. First, make them higher, and tell me what you expect to happen and why. Okay. So tell me already in the chat before you do any simulation, what to expect if you increase the J medium, the J between medium and the cell to a value higher than 10. More solid? Okay, why? Few fluctuations surface. Okay. Someone said the cell should shrink. Maybe a little bit. Okay, so let's do the simulation. Tell me if this happens. Okay, so if you do the simulation, you may notice that as you increase the addition value between the cell and the medium, the memory fluctuations decrease. And the bigger you make, the rounder it becomes, and the less you also reduce the movement and the cell shape change in the cell. So you all guess right what will happen. It will round and it become more rigid, okay? So this happens again because of the strongization of the contact surface between the cell and the medium. And once you have this value too high and you already is happy with this current settings, it will you will not accept any other pixel copy that deviates from its current value. So here in the simulation, you have a contact surface and a volume constraint. And because we want to minimize both the surface with keeping the volume the same, the resulting shape for your cell should be a circle or something that's very close to a circle. Okay. So once you get the circle with your desired that satisfy your volume, target volume, the cell tends to not change its shape anymore. And the higher you make this J, the more strong is this effect. So now let's do the other way around. Let's decrease the addition value between cell and the medium to lower values. Tell me what you expect to happen and why. Roundish rough surface. Okay. What if you keep decreasing it even more? Blabbing. Okay, so this is more or less what should happen. Okay, before you have 50, a very high value, it becomes very round. Now you start to decreasing, and the more you decrease, the less it cares about its current shape, uh, boundary outline, and start to become very rough. And if you actually make those j value equals to 0, that's equivalent of completely turning off the effects of constant energy, contact energy of the cell. The cell doesn't see the medium anymore. It doesn't see itself anymore. And when you make those j between medium and cell to be 0, actually turn off that plugin and you just fragment the cell, and the cell breaks into pieces. It becomes dust. Okay. So the lower the value of the j, the more easily it accepts deviations from its surface that it actually increase the surface of the cell with the medium. But if you decrease too much, you can actually lead to the fragmentation of the cell itself. Just like on the module before, you are decreasing the energy of the cells in the medium, and that you favor the cells to actually go to the medium and you fragment the tissue. Here you have the equivalent analogous situation where you actually fragment the cell itself. Okay. Does this make sense? Okay. So okay, but it's not the whole story about contact energy. There is another parameter that also plays a role in it. Okay. And this parameter is called the neighboring order. Okay. So remember that what we talked about contact energy before is that the way it works is every time you have a spin fluctual, the energy works by looking at that spin that has been flipped and looking all around its surface or its neighborhood, and then calculating all those j values between that pixel and all the pixels around. Okay. But the question is, okay, how you define this neighborhood at which you look around each one of those pixels? Okay. So in this case, you have this cell here, and you're flipping this pixel here, and then you're looking at all those other pixels around it and calculate the j between this and this, 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 and so on. Okay. Actually, these ones here do not calculate because they belong to the same cell, so you define them to have a 0 contribution, but you calculate these other ones. And then how you determine how far you go this? Should you go all to this or only to this 4 here? So there's a parameter in your contact energy plugin which is called the neighbor order. Okay. So there's a neighbor order which basically sets how far you look around. So the neighbor order is defined as following: is the set of sequence around a sequence of pixel sets around the center pixel. So neighbor order 1 means these first 4 pixels here in a 2-dimensional square lattice, of course. In a 3-dimensional is going to be different, and an hexagonal lattice will be different as well. So first neighbor order is just these 4 ones. Second neighbor order, you include those corners. Third neighbor order, you include these ones. Fourth neighbor order, you include these. And the total number of pixels which you actually use to calculate your contact energy term keeps increasing as you keep having higher and higher neighboring orders. Okay, so that's something. So again, for a neighbor order equals 4, you look at this very first 20 pixels around that pixel that's being flipped at the moment. Does this make sense? So,
excuse me, does increasing the neighbor order increase the computational cost?
Yes, a little bit, because you have to calculate more pairs.
Okay.
Yes, it does. So if you don't extend this much more in theory, let's maybe try to do this in practice to see if this how this works. So yes, increasing the pixel order does make it, yes, the cellular Potts model comes from the Potts model, that comes from the Ising model. So, and this energy term is a direct inheritance of the Ising model. Okay. So let's see how this works in practice. No, sorry, there was this other question. Does increase the neighbor order make it long range interaction? Yes, as you increase the neighboring order that you're using for the contact energy, you also have the fact that a cell that, if there's another cell that is here, you start to see some effect of the other cell as you get near the cell. Okay, so increase the neighboring order, allow two cells that are not really touching to already start interacting. Okay, so there is this effect. Okay, but in the simulator that you are using, you only have one cell, okay, in the medium, so you're only seeing the effect of on the cell shape. Okay, as we explore this parameter, you see where you may what one of the possible meanings of this parameter. Okay, so let's set up a simulation to explore this. Let's make all our parameters equal to the basics parameters. Temperature equals 10, target volume equals 1000, lambda volume 10, and let's keep the J of the cell and medium equals to 10 as well. And now let's try to directly change this parameter called neighbor order. Okay. So here, if you go to the contact plugin in the code, which should be lines 40 to 46, okay, there is this line here called neighbor order, which is currently at 4. Let's make this, let's try to make the simulation with higher and lower numbers of neighbors and see what happens. Okay, so as you do this exercise, please pay attention that there are two neighbor orders in the in our comp cell codes. One that appears in the contact plugin, but there's another one that appears in the pots. We are only dealing with the second one. Okay, we are only dealing with the neighbor order that appears under contact. Okay. Okay, so please go there and do a simulation with a higher and a lower neighboring order, and tell me what do you see. So what did you guys got? So many say that when they use lower numbers, the cell look rougher. And when you use a higher number, what happened? It becomes roundish. Okay. So that's exactly what should happen. Lower number orders, it looks more rough, okay, and higher looks more round, okay. And so that's, you can may notice that that's something that already James said. This is kind of equivalent of increasing and decreasing the additional energy, okay. So remember, if you make the additional energy lower, it becomes rougher. That's kind of similar to this. And you make the additional energy higher, it becomes like rounder. which is like increasing the neighboring order. And that makes a little bit sense because the higher the neighborhood that you use, the more pixel pairs you are comparing and summing up. So having a higher neighboring order, you are effectively making your addition energy to have a higher weight in your simulation. And when you make it shorter, you're at most calculating 4 pairs, right? So it's less J's that you're summing up in the calculation of your energy term. So that's why if you suddenly want to increase your neighborhood from 4 to 5 or 4 to 7, you may suddenly see that the cells change shape a lot because you're effectively increasing the weight of the additional energy on your simulation. And as I say this, you may think that, okay, those two things are equivalent, but they are not, okay? Increasing the J or decreasing the J is not equivalent as decreasing the neighbor order or increase the neighboring order, okay? They have different and important effects. You can already tell to see if you compare this simulation here with neighbor order equals 1 and this one with J equals 1, you see that they are not quite the same, okay? They look different, okay? Let me explain what is this, what's different, okay? So what's happening is that, okay, the neighbor order and the contact energies are not the same, okay? They have similar effects, but they're very different from each other, okay? To highlight those differences, let's do a set simulation where we compensate this change from the other, okay? So we can use this table here, and let's do the following. If we keep the neighbor order equals 4, okay, and J equals 10 as a reference, let's make the simulation with n neighbor order equals 1. So when you do the simulation with neighbor order equals 1, you are only calculating 4 pixel pairs, okay? So what you can do to compensate is actually increase the addition value by a factor of 10 times 20 by 4, okay? So we are decreasing from 20 pairs of pixels to 4 pairs of pixels, so by a factor of 5. So let's increase the J by also a factor of 5 to compensate. We can also do the other way. Let's make a neighbor order of 7. A neighbor of 7 has 36 cell pairs, so let's decrease your J by a factor of 20 by 36, 5.26. So in terms of how much energy you calculate for each spin flexure, when you do these adjustments, you're calculating the exact same energy, okay? So if the neighbor order and the addition energy were the same, you expect to see the same cell output, but you do not see the same cell output. You see different, okay? So please try to do those two simulations and compare to what you had before and tell me what those differences are. The J medium is not being used in the simulation because you only have one medium. So that parameter there has zero contribution on that simulation and actually usually has zero contribution on all simulations. So there is no simulation that actually use more than one background. Yeah, it didn't have any impact. Okay, so have you tried simulation? Okay. So what happens if for n equals 1 and a higher j? Can anyone tell me? Okay, we are already over time, so we're gonna just jump to the next slide. So you should get simulation that looks like this. Okay, so when you use a lower neighboring order, but you try to compensate by having an increased j, what happens is that you get a very squarish cell. Okay. So why this happens? This happens because in all simulations, the weight of the contact energy is exactly the same from one to another. But when you look at neighboring order equals 1, you are only minimizing the surface in the very first 4 pixels. Okay, so each pixel only see the curvature of its own pixel. Okay, so it, in other words, it doesn't see a curvature at all. So what's happening is the cell shape tends to align itself with the pixels of the lattice, an effect which is called lattice pinning in this type of simulations. Okay, so that's bad. You can compensate that by actually using a very high number of neighboring order. So when you look at high number order and you compensate by decreasing the j's, okay, the cells looks more round, okay. So and then the cells tend to be smoother, okay. So it tends to be a little bit rounded, is a better rounded shape. Maybe that exactly snapshot that I took was not very good, but that's the effect it has. So usually you're basically averaging, looking at a higher value here, and here you're looking just right here. So here, you are only look at the very immediate neighborhood, so you don't really smooth the cell. But here you're looking at a higher neighborhood, so you smooth the cell. Okay. So usually it's better to use higher neighboring orders, but the higher neighborhood you use, you have these two effects. First, you slow down your simulation because you're doing more computation per pixel flip. And the other thing is that you start to get interaction of cells with other cells that are not really touching each other. Okay, so that may be an undesirable effect of this. So you want to usually use as a lower neighboring order that you can get away with. And in 2D simulations, in a square lattice, a neighboring order of 4 is usually the best one to use. And from my experience, when you go to 3D simulations, a neighboring order of 5 is the best you can use. Okay. Yes, if the square shape at the beginning is more related to the initial condition, yes, it can be, but you also have this strong alignment of the peaks of the boundaries with the lattice, okay, and that's usually an undesirable effect. Okay. So let me see. Okay, now what I would have continued this by exploring the effects of the surface energy, but we are already 15 minutes over time, so I will stop here with only this thing. So these are really the core of your simulations. In all your simulations, you have the volume constraint. You always, in other simulations, you have the energy constraint, okay? And in all simulations, you are varying this parameter in respect to temperature. So this basically covers all the basics, okay? Maybe I can tell you about, we can discuss about the Surface plugin and the Length plus plugin. Or you can try by yourself. The slides are made available on the Google folder, and you can try by yourself late tonight or early tomorrow morning. Okay, I'll stop here, and again, thank you for your patience and going along. And you can do all the surface constraint and length constraint as homeworks.